Imagine holding in your hands the key to understanding life itself, only to watch others receive the glory for unlocking the door you opened. What you are about to discover reveals one of science's most profound injustices, a story that rewrites everything you thought you knew about the greatest biological breakthrough of the 20th century. In a dimly lit laboratory in London, a brilliant scientist worked tirelessly with X-ray crystallography, capturing images that would change our understanding of human existence forever, yet her name was nearly erased from history. This is the untold story of the woman whose groundbreaking work made possible one of humanity's most celebrated discoveries, only to have her contribution buried beneath layers of academic politics, gender discrimination, and scientific theft. Her name was Rosalind Franklin, and without her meticulous research and revolutionary photograph 51, the structure of DNA might have remained a mystery for decades longer. The year was 1952, and in the basement of King's College London, Franklin was perfecting techniques in X-ray diffraction that no one else in the world had mastered to such precision. Born in 1920 in a prominent British Jewish family, Franklin showed exceptional aptitude for science from an early age, excelling in physics, chemistry, and mathematics at a time when women were actively discouraged from pursuing scientific careers. Despite societal barriers and familial skepticism, she earned her doctorate from Cambridge University in 1945, focusing on the physical chemistry of coal and carbon, work that would prove invaluable in her later research. After spending several years in Paris mastering X-ray crystallography techniques at the Laboratoire Central des Services Chimiques de l'État, Franklin returned to England with skills that placed her at the forefront of molecular biology. She joined King's College London in January 1951, tasked with investigating the structure of DNA molecules using the most advanced imaging technology available at the time. If you find these hidden stories as fascinating as we do, subscribe to Hidden Minds and hit the notification bell so you never miss the untold tales of forgotten geniuses who shaped our world. Check the link in the description for additional resources about Rosalind Franklin and other overlooked pioneers whose contributions deserve recognition. Franklin's approach to science was methodical, precise, and uncompromising, qualities that would produce extraordinary results but also create friction in the male-dominated scientific establishment of mid-century Britain. Working alongside a PhD student named Raymond Gosling, she refined the X-ray diffraction technique to unprecedented levels of clarity, taking hundreds of images of DNA fibers under carefully controlled humidity conditions. What Franklin discovered through her painstaking work was that DNA existed in two forms, which she designated a DNA and B-DNA, depending on the relative humidity of the fiber samples. The B form, captured under higher humidity conditions, would prove to be the biologically relevant structure, and Franklin's images of this form were breathtakingly clear. In May 1952, after months of optimization and countless failed attempts, Franklin and Gosling captured what would become known as photograph 51, an image so clear and geometrically perfect that it practically revealed DNA's helical structure on its own. The photograph showed a distinctive X-shaped pattern, a hallmark of a helical structure, with clear evidence of the molecule's dimensions and the regular spacing of its components. Franklin's notebooks from this period reveal her methodical analysis of the data, calculating the precise measurements that would define the DNA double helix, including the diameter of the helix, the pitch of the spiral, and the spacing between base pairs. However, Franklin's scientific caution and perfectionism meant she wanted to gather more evidence before publishing definitive conclusions, a decision that would have devastating consequences for her legacy. Meanwhile, at Cambridge University's Cavendish Laboratory, two researchers named James Watson and Francis Crick were also racing to determine DNA's structure, though their approach was primarily theoretical rather than experimental. Watson and Crick were building physical models based on available data, trying different configurations to find one that fit with known chemical principles and existing experimental evidence. In January 1953, Without Franklin's knowledge or permission, her colleague Maurice Wilkins showed Watson photograph 51 during a visit to King's College. Watson later described this moment in his controversial memoir, admitting that seeing Franklin's photograph was the crucial breakthrough that allowed him and Crick to complete their DNA model. Additionally, Max Perutz, a senior scientist at Cambridge, provided Watson and Crick with a detailed report Franklin had written for a medical research council committee, giving them access to her precise measurements and analysis. Armed with Franklin's data and imagery, Watson and Crick rapidly refined their model, completing it in March 1953 and rushing to publish their findings in the journal Nature. Their paper, published on April 25, 1953, famously included the understated sentence describing the double helix structure and its implications for genetic replication. That same issue of Nature included supporting papers, including one by Franklin and Gosling that provided the experimental evidence confirming the Watson-Crick model, but their contribution was positioned as mere confirmation rather than foundational discovery. The presentation of the papers created the impression that Watson and Crick had independently deduced the structure, with Franklin's work serving only to validate their brilliant insight, when in reality her data and images were essential to their model's completion. Franklin, ever the rigorous scientist, never publicly complained about this appropriation of her work, instead continuing her research and moving to Birkbeck College in 1953 to work on tobacco mosaic virus structure. Her work at Birkbeck was equally groundbreaking, producing fundamental insights into viral structure and contributing significantly to the emerging field of structural virology. Tragically, Franklin's career was cut short when she was diagnosed with ovarian cancer in 1956, likely caused by her extensive exposure to X-ray radiation during her years of crystallographic work. She continued working throughout her illness, publishing 17 papers between her diagnosis and her death, displaying the same dedication and brilliance that had characterized her entire career. Rosalind Franklin died on April 16, 1958, at the age of just 37, leaving behind a legacy of rigorous scientific work and unanswered questions about recognition and credit. 4 years after her death, in 1962, the Nobel Prize in Physiology or Medicine was awarded to Watson, Crick, and Wilkins for their work on DNA structure. Franklin could not be honored posthumously under Nobel Prize rules, but even more troubling was the complete absence of acknowledgement of her crucial contribution in the award ceremony and subsequent celebrations. Watson's memoir, published in 1968 with the provocative title The Double Helix, portrayed Franklin in deeply unflattering terms, describing her as difficult, uncooperative, and unable to interpret her own data. His characterization of Franklin, whom he dismissively called Rosie despite her never using that nickname, revealed not just personal bias but the endemic sexism of scientific culture in the mid-20th century. It was only through the efforts of Franklin's close friend and colleague Anne Sayer, who published a biographical defense in 1975, that a more accurate picture of Franklin's personality and contributions began to emerge. Subsequent historical research has conclusively demonstrated that Franklin was not only a brilliant experimentalist but also drew correct conclusions from her data, conclusions that were appropriated without proper credit. Her laboratory notebooks, preserved at Churchill College, Cambridge, reveal that she had essentially solved the DNA structure independently and was preparing to publish when Watson and Crick rushed their model into print. The story of Rosalind Franklin is not merely about one woman's erasure from scientific history, it represents a systematic pattern of how contributions by women and minorities have been minimized or attributed to others throughout history. Recent decades have seen growing recognition of Franklin's essential role, with numerous scientific institutions and awards now bearing her name, including the Rosalind Franklin University of Medicine and Science in Chicago. In 2003, exactly 50 years after the publication of the DNA structure, The Royal Society established the Rosalind Franklin Award to recognize outstanding contributions to science, technology, engineering, and mathematics. Franklin's story has become emblematic of the hidden figures in science, those whose work was fundamental but whose recognition was denied due to prejudice, politics, or premature death. Modern scientists studying her original laboratory notebooks and photographs express amazement at the quality of her work and the sophistication of her analysis, often noting that her techniques were decades ahead of their time. The precision of photograph 51, which can now be analyzed with modern computational methods, reveals details that Franklin recorded in her notes but that weren't fully appreciated until decades later. Her contributions extend far beyond DNA, as her later work on virus structures laid foundations for understanding molecular biology and would prove essential in developing vaccines and treatments for viral diseases. Every time modern medicine uses our understanding of genetic structure to develop new treatments, diagnose diseases, or advance biotechnology, it builds upon the foundation that Rosalind Franklin's meticulous work established. The question remains why someone whose contributions were so fundamental could be so thoroughly erased from the narrative, and the answer lies in the intersection of gender discrimination, academic politics, and the Nobel Prize's prohibition on posthumous awards. Franklin's story teaches us that scientific progress is rarely the result of lone genius but rather builds upon the work of many contributors, some of whom history remembers and others who remain in the shadows. As we continue to uncover more stories of forgotten innovators and overlooked pioneers, Franklin's legacy serves as a reminder to question the narratives we've been told and to seek out the hidden minds behind every great discovery. The woman who captured the image that unlocked life's deepest secret deserves her place not as a footnote but as a central figure in one of humanity's greatest scientific achievements.